So, these are the lecture outlines. Biomaterial is defined as a substance that is going to become an integral part of the living system. So, it is a material which is engineered with great care so that it becomes part of the complex living system during the course of the process of the living being, be it in the form of a therapeutic event or as a diagnostic procedure. So, why do we need biomaterials, or why do we use biomaterials? Because these are naturally available and they can actually execute biological, mechanical and many other functions suitable to a living environment. And classification of biomaterials is that we have polymers, ceramics, semiconductor materials and metals. There are other classification systems of biomaterials as well. The most common ones are a wide variety of categorizing them as classical biomaterials, which actually goes down into subdividing into metals and alloys and non-metallic elements. So, these are the usually used material classification system, and then we have the nano phase biomaterial classification system. Here we have again metals and alloys and then non-phase metal, non-metallic alloys. So, this is how we go into classification. So, what are the considerations which are usually taken care of or thought of during biomaterial fabrication is that why is it required, and there is a proper specification for every biomaterial which is to be taken for designing, and an accurate characterization for that particular environment is very important, for which the biomaterial is desired to function, and that environment has to actually get integrated into this biomaterial and hence allow that biomaterial to carry out its function. So, this particular biomaterial has to also be very safe to the human being, and characteristics of a biomaterial is that it has to be non-toxic, it has to be biocompatible, and there should be absence of a foreign body reaction, and it also in addition has to possess the mechanical properties with good mechanical performance, mechanical durability and physical properties good enough to mimic the tissue which is going to be used for replacing the tissue in that particular environment. So, the first and foremost biomaterial is polymers, and these polymers are actually natural or synthetic macromolecules, which means that there is a bigger, larger molecule made of smaller monomers. So, we have a monomer that repeats to form a polymer. So, these are small monomers making into a larger polymer. So, finally, you have a macromolecule that is formed. So, this is a basic biomaterial which is actually used, and why do we use polymers is what we need to know. So, polymers are very convenient, so feasible. And why is it being feasible? Because of the versatility of the material. So, what do you mean by versatility? The meaning of versatility is that it can be tuned into, it can be blended into many properties, or it can be characterized with many multiple fabrication mechanisms, and it can be derived into various products good enough to suit an organ system, or it can be even for a diagnostic system. Whichever way it is, it can be molded or tuned into various requirements. That is the meaning of versatility. So, because of its very advantageous physicochemical, interfacial and biomimetic properties. So, because of this versatile nature of polymers, we have a huge range of applications which the polymers can offer. So, they have they can be used for making cardiac valves, artificial hearts, vascular grafts, breast prosthesis, dental materials, contact and intraocular lenses, fixtures of extracorporeal oxygenators, dialysis and plasmapheresis systems, coating materials for medical products, surgical materials and tissue adhesives. So, these are very important about polymer biomaterials or polymeric biomaterials. Polymer biomaterials can be divided into natural and synthetic. So, as we say natural and synthetic, we know that they are derived from natural products, and we have many number of times heard about the ones which are listed below. So, cellulose we know is a very important component of the living system, and then we have chitosan, which is a protein which are derived from insect based coating system, and then we have carrageenan. Carrageenan is actually a seaweed derivative, and then we have synthetic polymers. Again, synthetic polymers are further divided into degradable and non-degradable. As we see degradable, we can see that they are polylactic acid, polycaprolactone, polyurethane, polypropylene, PTFE and PET dacron. And then further we have non-degradable nylon. So, these are the classification system. Further, there is another method to classify polymers. Again, these are based on the characteristics of polymers or the basic properties of polymers. It is actually divided into three. The first one is elastomer, then we have thermoplastics and thermoset polymers. Then further, the first thing which we would be doing today is the natural polymers. So, polymers, as already mentioned, we know that collagen, then we have cellulose, chitosan, carrageenan and alginate again is a seaweed derivative, and then silk worm, the natural silk which is derived from the silkworm, has a beautiful polymer in it which is called as fibroin. This fibroin has been utilized in the recent times into multiple applications for many applications, and it is found to be very useful, especially in tissue engineering, bone and ligaments. The other systems like collagen are used mainly, most common usage are to make collagen sponges, where it is used to treat burns, wound healing. Collagen nanoparticles are also used for drug release applications as well. Then we have chitosan. Chitosan is actually being celebrated as a natural polymer, and we have enormous amounts of research work going on in chitosan. So, chitosan again is biocompatible, bioactive, biodegradable, and it also has antibacterial, anti-acidic and antimicrobial effects. So, that is actually a very characteristic or most sought after biomaterial right now. And then we have the seaweed derivatives, alginate and carrageenan. So, these are again very commonly used, mainly because of their very favorable characteristics and properties which is required for a biomaterial. Then we have synthetic polymers. We already know that synthetic polymers are divided into degradable and non-degradable. So, having that in mind, a quick look at the synthetic polymers. We have polyolefins, fluoropolymers, polyvinyl chloride, silicones, polyurethanes, polymethyl methacrylate, polyesters, polycarbonates and polyethers, and then we have polyamides. So, all these are actually comparatively are synthetically derived, and there is a very big range of applications which the synthetic polymers do offer. And because of their versatility, as already said, they can be used as medical devices for diagnosis and for therapeutic approaches. So, they are either used as a defect, as a filler in a defect, or they can be used as a stent, or they can be used as a valve, or they can be used as an adhesive. The applications are really very wide and multitude. And then we should definitely know, having said that it has got such a huge range of applications, we definitely need to know what are the advantages and what are the disadvantages. The advantage being ease of manufacture. As already mentioned, it is very easily available, and ease of secondary processability, because then you can actually do a secondary processing, and availability with desired mechanical and physical properties. Above all, it is very reasonable. So, it is cost effective. But the major disadvantage lies in the lack of biocompatibility in majority of cases, and hence their usage is often limited by the inflammatory reaction which is actually caused because of polymeric biomaterials. So, what happens? So, can we just ignore these polymers? Never. You can actually utilize them, but again combine them with other agents so that it will improve the biocompatibility, and so that there will be lesser inflammatory reactions. As already mentioned, polymers are classified into elastomers, thermoplastics and thermosets. The first one is elastomer. And these elastomers are actually special polymers that are very elastic. As the name suggests, it is very elastic, and they are tightly cross-linked and amorphous with a glass transition temperature well below the room temperature. The intermolecular forces between the polymer chains are rather weak, but this weakness is what actually gives that elasticity to the elastomer. And further, getting on to know further points about elastomers is that this elastomeric polymer, though it is actually elastic in nature because of the weak intermolecular bonds, it actually offers high toughness and excellent elongation, and before it could just break with before it can actually give up. So definitely it is a material which can offer lot of toughness, and these elastomers have very high molecular weight and they are amorphous in structure. So the moment we see amorphous, which means that they are not crystalline and they have weak inter and intramolecular forces. So these are the ones which are actually translating in low elastic modulus with high elongation. The glass transition temperatures are very important as we read polymers. So glass transition temperatures here in elastomer is below the room temperature, making them soft and flexible. So depends on the glass transition temperature, you can actually understand how the material would be at room temperature. And then is the description of how elastic is the elastomer. So, or can we modify it by further additions, or can we reverse the deformation and it can be further, the molecular bonds can be strengthened or not. So that can be done by addition of peroxides, metal oxides, sulfur, ionic moieties, or by radiation cross linkage. So this reversibility of deformation would require some presence of cross linkage. So how do we cross link those weak bonds? We cross link by adding peroxides, sulfides, metal oxides and radiation. And these also further, in addition to cross linking, can be further added on with filler materials. So those are again can be carbon, silica, clay, talc, mica are used as fillers for reinforcing elastomer. So first what we need to understand is elastomers are preferred because of its elasticity, but situation demands where we might want the toughness to increase. At that time we can add a cross-linking agent in addition, for if the further there is a reinforcement required, then further there can be fillers which can be added to elastomers. So this is a schematic diagram which actually represents the thermoplastic elastomer and thermoset elastomer. So we would go into the details of thermoplastics, we would understand that best. So thermoplastic, as the name indicates, has a meaning which suggests that there is a plasticity to that material which reacts with increase in temperature. So what exactly does thermoplastics mean? So it is a soft, it becomes soft over a specified temperature range. So it becomes plastic at a specific temperature, or it becomes soft at a particular temperature and becomes a solid again on cooling. So the examples of thermoplastics are your acrylonitrile, butadiene, styrene, nylon, polybenzimidazole, polyethylene, polystyrene, polyvinyl chloride and Teflon. And again, unlike elastomers, these polymers, that is your thermoplastics, do not require cross-linking because they have a presence of crystalline domain. At this particular juncture, we should please recollect elastomers were amorphous in nature. And next comes slightly more harder, your thermoplastics. So this thermoplastics does not require cross-linking because it actually has a crystalline domain, making it more tougher. So what happens as it becomes crystalline, the intermolecular bonding becomes more stronger. And presence of crystalline and amorphous domain together allows a wide range of thermal and mechanical properties, opacity and permeability. So there is a merger of two characteristics, that is, it is amorphous, at the same time it is crystalline as well. So the advantages of both the materials can be used up here. And please do note that the glass transition temperature of thermoplastic can be above or below the room temperature. That is because there is again a element of amorphous structures within. So, there is always the glass transition temperature is almost similar to your elastomer. So, thermoplastics can be actually extruded or injection molded or compression molded or transfer molded, whichever way we want to fabricate it. So, if in case if there is, I said cross linkage is not required, but in case if you want to reinforce the material, you can actually add nanoparticles to improve the properties like flame retardancy, durability and elastic modulus of thermoplastics. So, this picture over here actually shows that the link between the intermolecular link is slightly more stronger than what you see in elastomer. Now, we move on to the other end of the spectra of polymers. The first was elastomer, then we read about thermoplastics, and the next one is thermosetting polymers. So, in thermosetting polymers, the highly cross-linked polymers cannot be recycled or reprocessed, and they would be really solid. They cannot be reversed. There is no reversibility in thermosetting polymers. So, prior to cross-linking, they can be monomers or oligomers or even molten polymers, but then once cross-linking takes place, it becomes a solid. So, with a suitable initiator, a sensitizer or a radiation agent, this can become a highly rigid three-dimensional structure, making a thermosetting polymer. So, the examples of thermosetting polymers are your epoxy resins, polyurethane resins, acrylic resins, which are widely used thermosetting polymers. We have been hearing about all these resins quite in many applications. And what are the advantages of thermosetting polymers is that there is definite corrosion resistance, structural integrity because of its rigidity, low cost, thermal insulation and dielectric strengths of the thermosets, which makes them useful for many medical applications. Not only for any filler materials, it is also used for medical instrumentation, tools and prosthesis. And then we go on to reading about fillers. We have just completed polymers, and now we in between read that elastomers and thermoplastics require the presence of fillers. And what are these fillers? So, biomaterial fillers, what are these? So, as we add a biomaterial filler to a biomaterial, it makes the material more stronger, that is it reinforces, makes it more stronger, and then it allows the material, it gives more conductive ability, it also renders antibacterial effect to the material and other functionalities as well. So, a quick look at what are the fillers which are actually present. We have bio-based fillers and non-bio-based fillers. So, these are the broad category by which the fillers are classified. So, if you see the bio-based, again we have cellulose, natural fibers, graphene, flour, lignin and biochar. Here we have glass fibers, carbon fibers, graphene, talc, nanoclay and carbon nanotubes. So, these are the filler materials which are non-bio based. So, natural and artificial. So, natural, as the picture mentions over there. So, these are the commonly used fillers, the raw materials from which the natural fillers are derived from. So, all these are very useful in biomedical application, mainly because the single one term which is very important as far as biomaterial is concerned, that is biocompatibility. So, natural fillers obviously would definitely be biocompatible. The advantage of natural fillers is that you can get it easily, it is easily available, there is good mechanical properties, ease of processability, low cost, low density and biodegradable. That again is a very important characteristic which one looks for when selecting a biomaterial. And then the disadvantage are its mechanical characteristics. Density again varies with the source of origin. The other inorganic filler materials are that they are synthetic and they are actually more unique and they can exhibit electronic, optical, magnetic and mechanical properties. So, usually metal oxides are used as inorganic fillers, and these inorganic fillers are actually capable of giving out an electrostatic interaction. It can be also giving out a hydrogen bonding, and then it can also create a dipole-dipole interaction, making it very, very interesting and obviously giving in much more applications. So, the examples of inorganic fillers are your iron oxide. The moment we see iron oxides are of great use in diagnostic imaging, especially in magnetic resonance imaging, where it is most sought after metal oxide. And then we have gadolinium, that is used as because of its photoluminescence properties. And then we have gold. Gold has a very important characteristic that it can be used as a target agent, as a drug delivery system, and it also has affinity for many biological tissues. And then we have cerium, and these cerium and gadolinium nanoparticles are actually very importantly used as an antioxidant agents, and also they reduce mitochondrial damage, which is of great importance in metabolomic studies. And then further we have gold. Gold based nanoparticles are also used as a sensitizer in photothermal agents, and silver well known for its antimicrobial agents. A quick look at the fillers, the synthetic fillers. The most commonly used ones are your carbon nanotubes. As the name suggests, it is a tubular structure, and this is actually made of, or it is an allotrope of carbon, and it has a very high area, resistant to metabolism, aromatic structure. It has a tubular geometry, the shape is of a tube, and the nano size of diameter offers opportunity to deliver therapeutic or diagnostic molecules at the targeted site. So, it is very small, at the same time very highly durable, and they can also absorb and conjugate variety of drugs. So, that increases its efficacy and serves as a very effective drug delivery system. So, this is of great biomedical importance. And then we move on to graphene. Graphene again is a carbon allotrope. So, this graphene is actually a two-dimensional one atom thick sheet of carbon with a hexagonal conjugated structure. So, we can see that with this picture here. So, it has a plain D bond and plain P bond, and this graphene sheet has a exceptionally high mechanical strength, making it the most strongest material discovered. So, the most strongest material which is actually synthesized is actually graphene. So, because of these characteristics and because of the inherent properties which thin sheets of carbon can provide, they are used as biosensors for glucose, dopamine, DNA and proteins. And then we also have use it as a drug delivery system, medical imaging, photothermal therapy. Just look at how much of a variety of applications it can offer to us. So, these are again a wide variety of the carbon allotropes which can be used as fillers. The ones which we saw are the carbon nanotubes and the graphenes. In addition to that, we have numerous other carbon allotropes of great interest in biomaterial science. And then we go on to polymer blends and composite. As the name indicates, a composite means a mixture of two elements. Again, polymer blends, as the name indicates, there is some other material which is blend into polymer to derive a new material, so that there is an advantage. By mixing two material, we derive a synergistic advantage. So, a polymer blend or mixture is similar to a metallic alloy in which two polymers are combined to create a new material, the advantage being good biodegradability, hydrophilicity, mechanical properties, stimuli response, electric and magnetic properties. So, there are various types of polymer blends. We have heterogeneous compatible polymer blends and homogeneous polymer blends. So, as the name indicates, we can see that the heterogeneous things are immiscible and there is a heterogeneous tendency to it. It does not gel with each other. They stay together, but they do not gel. And then we have the polymer blends which are miscible. So, these two are important. And then further, to understand the applications of polymer blends and composites, again like how we saw for other biomaterials till now, these are also got numerous applications. So, like for example, you have your alumina, which is a blend of alumina and glass, and then we have it to be used for implants. Zirconia again implants and replacements, calcium phosphates in bone repair and augmentation, porcelain in dental restorations, and carbon in heart valves, percutaneous devices and dental implants. And this is a very big interesting list about polymer blends. These were so tempting that it had to be included in the list of polymer blends over there. This is just to give you an overview about how much of polymer blends are available and to give you an idea of how much of applications it has to offer. And looking at the scope in this particular list, it was so tempting that though there was a lot of text, it was so tempting that every learner of this course should benefit into the intricacies of this particular material. So, a quick look or a quick glance in these polymer blends and composites is that we have beautiful combinations being applied into multitude of a range of applications. So, it starts from being a filler material, it starts from being a graft material, it starts from being a drug delivery system, as a biosensor, as a cardiovascular stent, as a valve material, and then it is used as an antibacterial agent and so on. So, there is never ending opportunities, hemostats and so on.